Okay, so today we're going to do a demonstration of osmosis. So I've got a potato here to start with. So what I'm going to start by doing is cutting to roughly the same size pieces of potato to go in our sample. So I cut this here. So they should be roughly the same size. So I've got two rectangular pieces of potato here, which are both roughly about 6 centimetres in length. So they look the same here. The next thing that I'm going to do is I've got 10 mils of water that I'm going to put into my 2 wee tumblers. So get another 10 mils. 100 mils, sorry. And in this one I actually have some salt. Okay, so the water should dissolve the salt. Okay, so if I just swirl this around, you can see that the salt is starting to dissolve in that, and I'll give that a couple of seconds to go. So I've got roughly the same size of potato. We're going to pop these in these 2 different beakers, we'll say for this purpose, and we'll leave it maybe about an hour, and then we'll check back and see what has happened at the end of that time. Okay, so that's my potato in the beakers. Okay, so that's been just a bit over an hour since we left our wee potato slices in these beakers of water. Remember, it is the tap water, I should have said beforehand, but this was boiled tap water. That's just to try and purify the water a wee bit, and so that it's purer water basically. And B had the salt water in it. Okay, so what we want to just start by doing, as I said before, we're measuring these on kind of like the size they are. So these were around about 6 centimetres beforehand. You could also measure with scales, however I don't have any scales, but what you'd have to remember to do if you're doing that is before you put the potato in, you'd have to dab any excess water off it before you measure, you weigh the potato. Okay. So I'm looking at the one that's in the purest water just now, and we've got that just a little bit over 6 centimetres just now, so we've seen an increase in this one. If I look at the potato slice in the salt water, again I'm just dabbing any excess off of this, and what we see with this one is that it's about 5.5 centimetres, that one. Okay, so if you put them side by side, you can see that they have, this one is definitely smaller than this one. This is the one in the purer water, and this one in the salt water. Okay, so what does that actually mean then? So this whole concept is called osmosis. Osmosis is a form of passive transport, which means no energy is required during this process. Osmosis is the movement of water molecules from a region of high water concentration to a region of lower water concentration through a selectively permeable membrane. If we look at our plant cell here, so we have our plant cell. I've just simplified this. We've got the cell wall on the outside, cell membrane just inside the cell wall, and we've got a large vacuole here. If we're talking about potato cells, they're obviously plant cells, and so the cell wall just ensures that it keeps its shape. They keep this kind of rectangular structure here. The cell membrane allows entry and exit of different substances into the cell or out of the cell, and so hence selectively permeable membrane there. The vacuole contains a lot of foods, nutrients, waste, so things like water inside the vacuole as well. Okay, so if we have a look at potato A here, we can see that we've got our potato and we've got the tap water that has been boiled. Okay, we've got a lower water concentration inside the cell compared to a higher water concentration in the beaker of water. Okay, so what is going to happen there is you have water going into the potato cell because you have a region of higher water concentration in the beaker and a lower water concentration inside the potato, and so water enters from the beaker into the potato, into the plant cells here, and so you see an increase in weight or size of the potato. In beaker B then we have a potato slice in salt water. There's a higher water concentration inside the cell compared with a lower water concentration in the beaker of salt water, and so what's going to happen here is there's going to be water exit of the plant cells here into the beaker of water, and so the potato will lose mass or it will shrink. Okay, and essentially what's happening in plant cell A here is it's becoming something we call turgid. Okay, so what's happening in here is the contents within the plant cell, including the vacuole, which will swell, remember that holds water and nutrients, and the cytoplasm will swell against the cell wall. The cell wall keeps the structure of the cell, and so this swells up and we have water inside the potato and it would increase in mass. What happens in potato B, the plant cell in potato B, is the cell wall shrinks, the cell membrane rather shrinks away from the cell wall. Your vacuole is also shrinking because water is leaving the plant cell, and we would say that plant cell B becomes flaccid or plasmalized. And so this is the concept of, well, not the concept, this is the theory of osmosis. It's the movement of water molecules from a region of high water concentration to a region of low water concentration through a selectively permeable membrane, which obviously is our cell wall in this case because it's controlling the exit and entry of substances in the plant cell.